All right. Good afternoon, everyone. It's a pleasure to be presenting at Roche's workshop and share some of the efforts that we've embarked on with Mark and team over the past few months. My name is Aziz Al'Khafaji. I'm the Director of Molecular R&D at Broad Clinical Labs, and I lead the Methods Development Lab, where we focus on creating and deploying cutting-edge genomics tools. When Roche reached out with the specs of their new SBX platform, we were excited to deploy it in ambitious ways that could mark a paradigm shift for key activities that we have going on at the Broad. We identified two main areas in the RNA space. The first is high-scale multi-omic drug response profiling in multiplexed PRISM cell lines, and the second being ultra-high throughput single-cell RNA isoform sequencing with SBX. My main focus for this presentation will be on the single-cell project, which Hulan and Guoping have led with support from Brian. So just some very brief background. KRAS is a commonly mutated cancer driver and was previously considered an undruggable target until researchers developed small molecule inhibitors which could inhibit that aberrant signaling. Despite very promising clinical outcomes, therapeutic evasion, drug resistance is still a big challenge. And if we can actually understand these routes, these mechanisms of evasion, we can actually drug those, have combination therapies, and realize the immense kind of potential within these drugs, so rescue that potential. To tackle this problem, we partnered with the PRISM team at Broad, which has developed multiplexed cancer cell lines consisting of 500 unique co-cultured lines, which can be distinguished through sequencing of their expressed PRISM barcode. This is an incredible resource which allows us to efficiently profile drug effects across diverse cancers in a single experiment. So we treated the PRISM pools with our RAS inhibitors, BA2865 and RMC6236, along with our negative control DMSO and a positive control that elicits a strong transcriptional response, so panobinostat, this is an HDAC inhibitor. We performed single cell at the 3 and 6 hour time points, and then as well a survival assay after 5 days to understand the survival outcomes. With this experimental setup, we're able to actually link survival outcomes to early and intermediate kind of drug response. And the idea being, can we find these transient key intermediate states that allow for that ultimate survival? And those are potential targets that we can validate, and if proven true, that we can go after them with secondary compounds. And so our primary readouts are the PRISM barcode. Again, this helps us assign and identify the specific cell line that transcriptional profile is part of. We also, this is a multiomic single cell profiling, and so we actually captured protein expression through the protein text discovery panel, which gives us reports on greater than 320 protein expression targets. This is in the context of the 10x Flex single cell platform, which is a probe-based whole transcriptome readout on the GEM-X. And of course, we're sequencing this on the SBX platform, which gives us, for this experiment, 80 billion reads. The data was phenomenal. I believe it was Mark who said that this picture looks like a picture from the Hubble telescope. And indeed, this is a universe of data to explore. So I'm just going to go through some high-level metrics that we're all used to looking at with single cell. So here we had a median of about 1,000 cells per cell line. For the per cell metrics, we got about 28,000 UMIs per cell, 7,000 genes per cell. So the data was again very high quality. There's no obvious batch effect detected. There's actually no green pixels on this image. This is actually kind of the kind of co-clustering of are two different channels from single cell. So orange and blue kind of make green in our visual cortex. Just as a high-level sanity check, cell lines from the same tissues were more transcriptionally similar. So you can see on the bottom left, the skin cancer cell lines, the top colon cancer and kidney cancer, et cetera. We also pseudo-bulked the different cancer cell lines from the single-cell data and compared that to orthogonally derived bulk RNA-seq, and they were highly correlated. So we just have very strong confidence in the quality of this single-cell RNA-seq data. Equally so, the protein expression data was very impressive. We're able to resolve many cell lines from protein expression data alone, which is pretty radical. And for the metrics there, we see almost 13,000 protein UMIs per cell and a median of 300 proteins detected per cell. So this slide underscores the utility of capturing both RNA and protein. So some RNA and protein expression is highly correlated, as you can see on the top right graph, and then on the bottom right graph, some of it is poorly so. So each dot in that graph is a cell line. The x-axis is RNA expression. The y-axis is protein expression from the panel. So you can see for a housekeeping like gene, it's highly concordant, right? But for things like transcription factors that are highly post-translationally regulated, i.e. protein degradation, if those switches are turned on and off, you're going to see a lot of difference there. So RNA is not always a good proxy for what's going on at the protein level, especially transcription factors, obviously very important. So this just highlights the power of capturing both of these modalities. The graph on the left just shows the range of correlation expression across cell lines, and so there's a lot of that variance. Okay, let's dive into drug response. As expected, we observed a range of transcriptional responses to our inhibitors. So annotating cells by treatment, we can use co-localization to get just a very high-level first glimpse at the various responses. Here, we can see that the treatments are not co-localized with our negative control DMSO, which is in purple. And this shows us that this is an example of a large transcriptional response to the inhibitors. Here, DMSO and RMC treatments are co-localized, so pointing to less of an effect on this cell line with the RMC drug. Vice versa for the BI drug. And here's a case where it looks like this line was not responding to either inhibitor, right? Okay, shifting focus now to the 5-day survival data, where dark blue is sensitive and yellow denotes like more resistant cell lines. I'll highlight 4 main classes that these cell lines fall into. Starting on the very left, this is resistant cell lines that are sensitive to both compounds. Just to the right, sensitive to RMC. Furthest to the right, sensitive to BI only, and then in the middle, resistant to both, right? In the interest of time, I'll just show that there are unique enrichments of mutation in these different classes, many of which are concordant with the literature. So in the sensitive to both category, we can see that that KRAS is actually enriched for, which is entirely in line with the mechanism of the drug. These drugs target mutated KRAS. And then we see different enrichments for various mutations for the uniquely sensitive cell lines. And then for the resistant to both, we can see that BRAF is one of the main enriched mutations, which makes total sense. This sits downstream of KRAS and does not need kind of this aberrant signaling from a mutated RAS to have its oncogenic effects. So this data was produced a little more than a month ago. So of course, this is just the beginning. From this first look, it's clear that there's a lot to discover from this data set. And we're excited to bring to bear these classical analyses, advanced AI approaches, and we will surface some of the complex biological signals. And we feel like this is a high-yield approach to systematically identify mechanisms of resistance, to be able to validate those, target them, and I think really does mark a paradigm shift for drug development. Okay, great. Now I'll shift gears, and we'll briefly touch on our efforts in the RNA isoform sequencing space with SBX. This work was led by Christophe, Brian, and Hulin. So we ran kind of like a traditional assay, PBMCs, and a 10x 5' gene expression kit, and we sequenced the pre-fragmented full-length cDNA libraries. We were able to resolve the canonical cell types and their biomarker genes. Everything was kind of nicely clustered. But we wanted to actually dive in and see what these read lengths look like and their ability to resolve isoforms. So on the far right is the read length distribution, the lion's share of which is between 500 and 800 base pairs, and you can see a fairly long tail heading to the right there. To understand the impact of read length on our ability to resolve isoforms, we informatically binned those reads from 300 to 1200 and 100 base pair increments. And then we ran this kind of isoform completeness metric called these categories called full splice match and incomplete splice match. We can see as we go from the 300 base pair bin to the 500 base pair bin, there's a sharp increase in percentage of reads that are full splice match. So that's a good quality metric for isoforms, right? And so this gave us a lot of confidence that these read lengths could span multiple splice junctions and even resolve full-length isoforms. And this is just a read level example of just that. We have the CD1C gene, and we see those reads spanning the multiple exons. The bottom two are known isoforms, and the top one, we can see that second to the last exon missing. It's not an annotated isoform, so that's pretty exciting. So I'd like to acknowledge the immensely talented scientists in my group, the PRISM team, Broad Clinical Labs, and our industry partners. The single-cell experiment in particular was an intensive effort, and it took everyone coming together to make it a success. And it's been a sincere pleasure collaborating with Roche on this transformational technology. And Mark, congratulations on this landmark achievement.